All right, so I want to explain one type of specific immune response to you called the humoral response. This one is also known as the B cell response. So it's a three-step process. In your body, you have your specialized white blood cells, your lymphocytes, which are called B cells. So that's an example of a B cell in the middle there. And as you can see, on the outside of B cells, they have antibodies, which act as their cell receptors. Now, what a B cell does is, once activated, it produces a number of other cells called memory cells and plasma cells. Let me explain the process. In your body, you have inactive B cells floating around. And to activate a B cell, well, we can do it two ways. One way we can activate the B cell is when an antigen from a pathogen actually attaches to the B cell. So the B cell will have a complementary antibody that's specific to that antigen. And when the antigen attaches, that will activate our B cell. The other way to activate our B cell is through what's called cytokines, which are released by helper T cells. Now, a helper T cell is another type of lymphocyte, which you would have learned about already. And what happens is an antigen presenting cell, which is a phagocyte, will actually engulf a pathogen. And when it does this, it will display the pathogen's antigens on its own cell surface. And our T helper cells attach to those antigens. And when they attach, they become active and they release these little proteins called cytokines. And the cytokines can also activate our B cell. Now, the process of the B cell attaching to the antigen and becoming activated is what we call step one of the humoral response, which is clonal selection. So clonal selection, the B cell attaches to a specific antigen and becomes activated. And the next step is that the B cell actually undergoes what's called clonal expansion. And in clonal expansion, that one B cell proliferates and divides multiple times to produce a colony of identical B cells. Now, these B cells which we've produced will have the exact same antibodies as the original B cell, which means they're all going to be specific to that same antigen, and they can also work to help bring that infection under control. So clonal expansion is when the B cell proliferates multiple times to produce a large colony of identical cells. Once we've undergone clonal expansion, we have a process called differentiation. And in B cell differentiation, these cells which we've now produced can divide into one of two cells. They can divide into what are called B memory cells. And your B memory cells are the cells that give you immunity, and they will remain in your body in case you come across that particular antigen for a second time, and they will enable your body to have a faster response, and that response will also be of greater magnitude. If the cells don't differentiate into B memory cells, they'll differentiate into these cells here called plasma cells, and the role of plasma cells is to produce antibodies. And the antibodies produced by the plasma cells travel through the body in the bloodstream, and these antibodies attach to the antigens on the specific pathogen to do, well, three things pretty much. So the antibodies, once they attach to the pathogen, they can cause neutralization of the pathogen, which means that pathogen will no longer be able to infect its host. They can also attach to the pathogen to cause opsonization, which makes the pathogen more identifiable, to phagocytes, which can come along and engulf the pathogen to destroy it. The third thing antibodies can do when they attach to pathogens is they can cause agglutination. And what this is it causes the pathogens to clump together. And then when a phagocyte comes along to engulf them, well, instead of just engulfing one, it can engulf a whole clump of the pathogens to help get rid of them from the body. So that's your B cell response. Step 1, clonal selection. The B cell attaches to the antigen and that can activate the B cell. And also the antigen presenting cell, displaying the antigen on its surface, activates a T helper cell to release cytokines, which also activates the B cell. Once activated, the B cell undergoes clonal expansion and produces a large colony of identical B cells. Those B cells differentiate into two types of cells: our memory cells, which stay in the body to give us immunity, and our plasma cells, which produce antibodies, which attach to pathogens to neutralise them, make them more identifiable, clump them together, so ultimately our phagocytes can come along, engulf them, destroy them, and get rid of them from our body.